On the science end of this, we're doing our darndest. People, thousands and thousands of researchers all over the world are doing their best to try and figure out what's going on with these Alzheimer's disease and these other dementias. And once we can figure out exactly what's going on, we can figure out how to slow it down and maybe even stop it eventually. So the best you can do now is to stay up to date on the latest developments and empower yourself, right? Because there are ways, as I'm going to mention here in a little bit, whether you are in your middle ages and still cognitively fine, or whether you're just starting to have memory troubles, or maybe are in even initial stages of dementia, there are things you can do to continue to protect your brain and keep things at bay. So just briefly, we're going to go through some brain basics just to kind of go over some key concepts to kind of keep in your mind going through all this. Talk about Alzheimer's disease a little bit, and we're not going to really speak much to some of the other causes of dementia, like vascular causes and things like that, but just more on AD tonight, because I want to go into the latest, the therapies that we have, and sort of give you some updates on what these latest drugs are all about. And then again, like I said, talk about these risk factors and, short of a treatment, what can we do to reduce our risk? And again, this is risk reduction for folks like me in, middle ages, still fairly okay. And then also for people who are starting to feel like their memory's declining. When we talk about just some brain basics here, right? So the lower right, you see the human brain, we think of it basically weighs 3 pounds, has 3 parts. You've got the cerebrum, that's the big part that we usually think about with the brain, right? And it's divided into what we call lobes, okay? And that's a structural division, but also a functional division, okay? So you see you've got your frontal lobe, which is up here, basically right behind your forehead. That's involved in what we call executive function, which is like, making decisions and plans and, just performing our jobs and taking care of things at home and things like that, right? But it's also sort of our personality area as well. Then we have the temporal lobe. Now, this is an area that's important in most dementias because it's basically right behind your ears, okay? And that's our memory and language center. So that's where we're storing new memories and retrieving memories and things like that. And then in the top, sort of at the crown of your head, there's the parietal lobe. And that's sort of the area that takes all the sensory input that you're experiencing right even at this moment, and then just sort of orienting you to what you need to do to take an action next with all this information that you have coming at you, okay? And then finally we have in the very back of your head, interestingly enough, is the occipital lobe, which is your visual center. So your eyes actually send signals to the back of your head, to the back of your brain, and that area tells you what it is you're looking at, okay? And then we have 2 other areas. We also have the cerebellum, which basically means little cerebrum, and that's our balance and coordination center. And then the brainstem, which is connected to the spinal cord, goes down, and that's our area that controls what we call autonomic function, which are things like our breathing and heart rate, things like that, as well as areas that help us to pay attention to our surroundings. Okay. Now, if we take a human brain and we slice it really thin and put it on like a microscope slide, right, and use a special stain, what we'll see are the cells of the brain, the main cells, and those are called neurons. So you see this nice little, right there in the middle, this nice little sort of round body in the middle, but then all these nice branches coming off of it. They're really pretty to look at under the microscope. And we imagine, so we sort of imagine this, because of all these sort of branches, we sort of think about a neuron forest in the brain, okay? And these neurons talk to each other by using special connections called synapses, okay? So they send chemical messengers across this little gap between the 2 cells, and they talk to each other, and then that neuron talks to the next neuron, and so on and so on, and then the next thing you know, you're remembering something or performing a task or what have you, okay? And so just kind of keep in mind these basic terms of lobes and neurons and synapses, all right? So let's briefly talk about Alzheimer's disease. So this is actually a picture of Dr. Alzheimer and his first patient, the one that he described, that formed the basis for the disease to eventually become named after him. Her name was August Dieter. It turns out she was actually very young. She was in her early 50s. And what we knew now is that she actually had a genetic form of Alzheimer's disease, which is a very, it's a very aggressive form, and it's just nonstop progression. Even the risk factors I'm going to tell you about don't seem to have any effect, okay? And they're very, very rare too, like maybe less than 5% of cases. And it really, it typically just happens to very young people, okay? But what I put up these other two names, Dr. Oscar Fisher and Dr. Solomon Fuller, because about the same time, this is around 1908, 09, 1910, more than just one person, Dr. Alzheimer, were actually, studying these patients and looking at their brains and looking at the pathology and trying to figure out what was going on with them. So Dr. Fisher was actually, he was a German guy, but he was actually up in Czechoslovakia, I think. But then Dr. Fuller was African-American, and he was at Clark University near Boston. And he actually gave a talk in 1908 about the same time Alzheimer did. It was at an international psychiatry conference, and this was attended by Sigmund Freud and Ernest Rutherford, who was actually the father of modern chemistry. So if you've ever had to sit through chemistry class, you can blame him. And again, we now know that the disease what differentiates Alzheimer's disease is that it begins with that memory impairment, and then as it progresses, you get into some language difficulties and just some trouble with activities of daily living and things like that. Okay, and so just from the sort of eyeballology level. So what we actually see is just sort of, and again, this is a severe brain here. So you see sort of the shrinkage of the brain, right? So that's kind of what you see at the end, especially this area called the hippocampus, which stores new memories. And so what really happens, if you look at the little orange cartoon there at the bottom left, is you have your nice little neuron with all the branches and everything, and eventually they just start to sort of die off, and then they disconnect. So they're no longer talking to each other. They lose those synapses. Okay. And in Alzheimer's disease, this happens again in areas of the brain that control memory and attention, things like that. Little pathology, because this is important for some of our drugs we're going to talk about. So you probably heard of the two pathologies, or plaques and tangles, right? And so the plaques, and this is actually a drawing from Dr. Alzheimer. He called them thickened miliary foci. But what we know now is that they're out in between those neurons, out in the forest, if you will, right? And they're made of these aggregates of a protein that's called amyloid, okay? And a lot of these new drugs are designed to target these plaques, okay? The idea is you get rid of the plaques, you can slow down the cognitive decline. And that's actually a picture that Dr. Fisher drew of the plaques about the same time Dr. Alzheimer was doing his drawings, and Dr. Alzheimer arguably the better artist there. And same when you compare it to Dr. Fuller's drawings of the tangles. Anyway, but the point is they were finding these things at the same time. It just, Dr. Alzheimer got, I guess infamous because the disease became named after him. Because we also get these tangles, and these actually have built up inside of the neurons, okay? And we know now, and so if you look at this picture over here on the right, all those little brown cells that are stained, that's abnormal. Those shouldn't be stained, because what that means is they have these tangles inside of them, okay? And we know now that these tangles are made of a protein called tau, and this will come back. So we got the amyloid for the plaques and the tau for the tangles. Okay. And again, these are Dr. Fuller's drawings of some of those tangles in the brains he was studying up in Boston. So the treatments, where are we now? Okay. Well, the old guard, right? So we've heard about, so these drugs have been around for over 20 years, okay? And they just treat symptoms. They're not doing anything to slow things down, really. But they can help. It just depends on the person. So they're basically two main drugs. They're what the so-called cholinesterase inhibitors. And the next slide, I'm going to kind of explain what that is, because those are sort of the main drugs that are being used. And then also what we call these NMDA receptor antagonists. And those, again, I'm not going to talk about that so much. It's the drug Namenda, excuse me. And it's just a drug that sort of helps protect some of the neurons that are like in the hippocampus where your memory center is and things like that. There's a newer drug where they're combining them, and this is called Namzanir. Yeah, it's the one of the cholinesterase inhibitors, donepezil, plus this memantine. Okay, and my mother-in-law actually has some dementia that's related to vascular. She had some strokes. Okay, and this drug's actually helping her. And so she doesn't. There's not even evidence that she necessarily has Alzheimer's disease. So again, if there's some other form of dementia that your doctor suspects, you can always ask about using these drugs, even though they're prescribed for Alzheimer's disease, because there may be some benefit to them. And then when we look at sort of the good and the bad here, so this now we're looking at sort of our bottom figure there, and I know it's probably hard to see from the back. I apologize, but basically, with the cholinesterase inhibitors, the good is that you get this increase in interest in daily activities. So people sort of just become more engaged, and once you become more engaged, you start to, again, it helps with your memory because now you're sort of making that effort to be in the present. But there are these side effects, such as rhinorrhea, which is the world's fanciest name of saying runny nose, as well as bradycardia, which is again, it just means irregular heartbeats, all that means. And then some nausea and some, diarrhea, things like that. And it can also cause sleep disturbances. So again, if you or someone or a loved one or if you're a care partner for someone, after a couple months, you need to sort of monitor how these drugs are doing with respect to these side effects too, in addition to trying to see if they're actually having some sort of benefit, right? And so just going into the cholinesterase inhibitors a little more closely, what are these drugs actually doing, okay? So what I'm going to do is start at the top right and then sort of work my way around the horn here, okay? So that little blue circle or little blue bean-shaped thing, right, in the drawing at the top left, that's the center for these neurons that are called cholinergic neurons, okay? And they provide a chemical called acetylcholine to all the lobes of the brain, okay? It goes to all the lobes. So it just sort of comes out. It's basically kind of like right here under the roof of your mouth is kind of where it is in the brain, but it sends out those branches all throughout the brain. And the reason this acetylcholine is important is because that's one of those chemical messengers that are used in the synapses so that the neurons use to talk to each other. And it just so happens that that chemical messenger helps us to pay attention and remember things, okay? What happens, though, is that during Alzheimer's disease, these cholinergic neurons, if you can just kind of look at, as you go in that black and white photos there, this is microscope slides using a special stain to see those cholinergic neurons, and you can sort of appreciate that as you go from the top picture there is a cognitively normal person, and then the picture at the bottom is someone with end-stage, Alzheimer's disease. So there's a good bit of loss of these neurons as the disease progresses, right? And so what that means is when those neurons are lost, the acetylcholine gets lost, okay? And so now you're losing that chemical messenger. What the cholinesterase inhibitor does, it keeps the acetylcholine active longer. So you've got less of it, but this drug's going to help what you have there work longer. So that's going to help you, again, help you pay attention and remember things better. So that's basically what these drugs do. So that's just, just so you know, I have some information about exactly what this is sort of the major thing that's been prescribed for the last 20 years or so. So this is the point of them, basically. But what about these new treatments? And what we're doing, these new treatments are targeting those amyloid plaques, right, just to slow down the disease, all right? And again, here's our amyloid plaque refresher. So again, those little gumball things there in the picture on the left, those are the plaques, right? And again, they're made of that amyloid or A beta, as they call it. And then what we can do is we actually can do PET scans and look at amyloid in the brains of living subjects, okay? So just like you would do a PET scan to visualize, say, a tumor or something, you can also use a different tool that goes up into the brain and it attaches to these plaques, and then you can visualize them in the brain as in a living person, which is really cool. And you can see, so for instance, you have like it's sort of in the middle here, you've got a normal person who's 83. And so when you have those like cool colors, that sort of blues, that means there's really not much there. But then you have another person, so down at the very bottom there, with dementia, and this would be pretty severe. You see these real sort of hot colors like the reds and the purples and things like that, right? Or pinks or whatever. And so again, it enables us to sort of track the amount of amyloid in a person without having to, again, like do a brain biopsy or something like that, okay? So this new drug Aduhelm that sort of is, for all intents and purposes, dead in the water, but you may remember it from a couple years ago, okay? And the drug is called aducanumab, and so what it is it's an antibody. This new class of drugs are all antibodies, okay? And you can think about, of course, think about antibodies sort of in the context of, for instance, COVID. Remember, we thought the whole thing was if you got infected with COVID, your body would build up antibodies to it, and then those antibodies would be like, heat-seeking missiles trying to find the COVID and attack it and get rid of it, okay? So what we've done here, what the scientists have done, is they've made a homemade antibody, if you will, that acts as sort of a heat-seeking missile to find the amyloid in the brain and the plaques and attack the amyloid and get rid of it, okay? And so if you look up at the top right there, what that's showing is this is 4 different people at baseline and then a year later, okay? And so the person on the top had the placebo, so they didn't get this antibody at all, right? And as you can see, they have about the same amount of amyloid in their brains. They've still got all those big red hot spots and everything, right? Now, if you go down, so what the next thing is showing is a person is 3 people who had sort of a low, medium, and high dose of the amyloid drug. And what you can see is a year later, you lose all those hot spots. So that means those plaques are actually going away. So the amyloid's actually finding those plaques, attacking them, and getting rid of them, okay? The question is there a clinical benefit to this? So if you remove the plaques, does it mean you get better? Well, interesting story. So this was, these were, and again, we're not going to dwell on, what these graphs, the whole nitty-gritty of these graphs. But what they're basically showing is they basically had 2 big trials they were running at the same time, okay? One was called the EMERGE trial, one was called the ENGAGE trial, okay? Now, the EMERGE trial showed 20% less decline in cognitive abilities with that highest dose after 2 years, okay? But an exact identical trial run at other hospitals and things like that showed no effect at all. So basically, you've got one that worked and one that didn't, and the one that worked was only like, it's not really clear what a 20% benefit is, right? So the other problem with this is that there were these side effects, okay? And so this is just the terminology that they use in the field. It's this, what we call ARIA, A-R-I-A. And what that stands for is amyloid-related imaging abnormality. And that's not really important. What's important is what it represents. And what it represents is brain swelling and brain bleeding, okay? And so what they found was that 35% of the patients showed swelling or bleeding. So that was not a good thing, right? And so the FDA granted accelerated approval anyway. And their rationale was that it was for, obviously a serious illness that's an unmet medical need. They felt like it was reasonable to predict that lowering the plaques, like we saw with the PET scans, would provide a clinical benefit. But what they required was another trial. And we want a bigger trial, a longer trial. Let's make sure, and if it doesn't work, we're going to take it off the market, okay? Well, it's kind of too late for it anyway, because most medical centers aren't even, especially including all the ones in Michigan, aren't even offering the drug. Medicare is only, it's a very expensive drug, okay? Medicare is only going to cover it if you're in this other trial. So that's like 1,000 people that are going to get coverage, and the rest of us would have to pay a whole lot of money. Plus, you have to pay the hospital to sit in an infusion clinic for an hour every month, and so it really adds up, and it becomes a socioeconomic inequity issue too, because basically now it becomes a drug basically for people who can afford it, right? And because of all these restrictions and costs and its questionable efficacy, it's really barely used in the US, okay? But along came the next one, and this was called lecanemab, and this one was given accelerated approval just this year, and it's the same kind of drug, okay? It's you're putting the amyloid into your bloodstream, this antibody into your bloodstream, goes up into your brain, finds those plaques, attacks them, and gets rid of them. Question is, again, is there a clinical benefit, right? And so what we found, or what they found, not me, was that what this graph is showing you here at the bottom is the lower that little line there, that's the more the amyloid is going away, right? So that black line at the top, that's the placebo. So again, the people who are just getting the sugar pill, so to speak, their amyloid's not going away at all over the course of 18 months. But the people at the high dose, that green line, they're getting a lot of reduction of the amyloid. So the same kind of effect they saw with the other drug. The difference is, this time what they found was that there was actually a 27% slowing of cognitive decline over 18 months. And so if you look at the little graph there, you see where it says worsening, right? So the people with the placebo got worse faster. So, you see the blue line is below that yellow line, and the yellow line are the people that had the drug. So they're getting worse more slowly, but also what they were seeing is that they're starting to get some separation, right? So that the space between those two lines is getting bigger as you get to 18 months. So maybe like 2 years, you get even better benefit. So that's where we're at with all this. And the other thing is that there are fewer side effects, right? So if you look at the very bottom, that little table there at the bottom, the very bottom line, remember there was a 35% side effects with the other drug. With this drug, it's only about 12 to 17%. So, about a third, which is pretty good. And so we'll know if this drug is going to be approved for get the full FDA approval basically in a couple months. And then after that, if it does, then, Medicare, Medicaid might actually cover it, but we don't know yet. The problem, of course, is this a here we go again. So we're starting to get these reports of people who are taking the drug and they're having these, again, this is a picture of a blood vessel in a person who died from when they were taking the drug. And that orange in there, that's the amyloid. And then that sort of yellow-green area in the sort of right there in the middle, that was where the vessel sort of broke and started to bleed. Okay. And again, this is one person, right? And every drug that we take has side effects, and especially when you have, think about chemotherapy. I've taken chemotherapy. I mean, basically, you're eating poison, but you're trying to get rid of the tumor. But there are side effects, right? And this is potentially one of them. And then the other thing that they're finding, and this actually was just published like last week or so. So was this one on the left. Was that it looks like there's some brain volume loss for people who are taking the drug. Now, the explanation for this is that it's happening because you're getting rid of the amyloid, and the amyloid's flushing out, so it's sort of shrinking the brain back. But people still aren't really clear what this means, because normally you would think your brain shrinking a little bit would not be a good thing. So there's still a lot of questions for this particular drug. But again, the fact that for the first time, it looks like there's something that could provide almost 30% slowing of decline over 18 months is very compelling, and that's really the best we've done so far. It's maybe underwhelming, but considering how much time and effort and money has gone into this, to finally see something that seems to maybe work is it's a win. But what about tangles? Now, this is actually just reported out a couple weeks ago. There's a new drug, and it's called B2B80 or something like this, right? And what it does is it's a custom designed molecule that lowers the tau levels. So again, remember our tangles are made of that tau, right, on the left. And so what it does is it takes away the tau, and then what it will do, at least in animal models of the disease, is it will remove the tangles and also improve their little ability to run through all the little mazes correctly, okay? And this was actually just published. And I just basically cut and paste the caption from the report. And what you can see here, these are two people, one on top of the other here, with mild AD, and you can see the tangles there, those little red hot spots. And what you can appreciate if you look down at the end is that after a year or so of this treatment, those little hot spots go away. So it's actually reducing the tangles. And this was a phase 1 trial just to make sure it was safe. And this was, actually this was just reported at what's called the AD/PD conference, Alzheimer's disease, Parkinson's disease, which is a big international conference. And so that got people excited. The next thing they're going to do with this then is take a phase 2 trial and start treating people and see if it's going to improve cognition as well as lowering the tangles. Okay? All right. What else is in the pipeline? I'll be brief because I realize I'm rambling a little bit too much here. Right now, there's a Professor Jeff Cummings who does this every year. He sort of does like a roundup of all the clinical trials that are out there. And what we're seeing just at the top here is that right now there are 31 drugs that are in phase 3. So that's thousands of subjects being tested over like 2 years or so. Okay, so these are pipeline drugs. Some of them are to treat neuropsychiatric symptoms. Some of them are just for cognitive enhancement, right? But about 70% of them are so-called disease modifying, meaning they're designed to slow the disease down, okay? And of those, if you kind of see the little wheel there on the far right, you see there, you've got on the far right, you've got your amyloid drugs, but then we've got, again, drugs against tau and some other things like inflammation and vascular problems, and even to try and help the synapses to work better, things like that, okay? And then if you look at the bottom, there are actually 82 agents in phase 2. So there are all these, so, even if a third of those seem to work, now we've got, a good 30 more drugs that can go into phase 3. And so eventually something's gonna give, I'm sure. But in the meantime, we're trying our best. We're maybe seeing a little bit of glimmer, with these amyloid drugs, and obviously these other drugs that are in the pipeline. What can we do now to reduce our risk? Okay, and again, I want to stress this is not just if you're healthy and worried about protecting yourself. This is also if you're having memory troubles, and it's just going to these things will also help slow things down. Okay, or even maybe just stop them in their tracks. And so, since it's now spring, of course it snowed this morning, but, whatever. We can get out and walk more, right? And so this was reported last year after our talk, our research update. And this was the EXERT trial. And these were 300 people with mild cognitive impairment, and they were in, they were in 2 groups. They either aerobic exercise or just sort of stretching and balance and range of motion. And then they also had a group that didn't exercise for comparison. And what they found there, the bottom bullet point, is that the 12-month update, so they just sort of did an interim analysis, they already found that the exercise groups were showing no cognitive decline, okay, compared to, whereas the control group showed significant cognitive decline, the people with mild cognitive impairment, okay? So again, just exercising. This literally just came out, well, it was last year, but it was again after our talk last year. Just walking, okay? These steps add up. This is a study in the UK of almost 80,000 people, okay? And their average age was 61, okay? And they were giving these little step counting wristbands, and they were tracked for 7 years. And what this graph is showing you is, so you can see the total steps, so you see the total steps right there along the bottom, but then declining risk is when that line is going down, okay? And so what you see is that there's this peak protection at like 10,000 steps a day, which admittedly is a lot, okay? That's like 5 miles. So, the good news is if you walk even more than that, it starts to go away, so don't overdo it, right? But the cool thing about this was they started to see a beneficial effect at about 3,000 steps. So my wife, I was reading this, and my wife has one of those, Garmin watches and things like, she likes gadgets. I can barely work my iPad, but so we were like, let's walk the dog. Let's see how many steps we actually do. So we walked the dog for about 30 minutes. We went grocery shopping. Just whatever. Just by the end of the day, we had gone about 6,000 steps. So that's just walking around for 30 minutes and going shopping and just doing your thing. So there you go. So our other thing, of course, is diet. we've all heard about the Mediterranean diets, eating the fruits and vegetables and fish and poultry, and lots of studies showing that that's protective. And these are with thousands and thousands of people that have gone through these tests and all over the world, and they all show a positive benefit, about a 30% protection. Okay. This kind of turned this on its head. This is a Brazilian study of 10,000 people. And what they did is they compared rates of cognitive decline in people who consumed high amounts of ultra-processed food compared to people who did not consume a lot of processed food. So what do they mean by processed food? Basically, things that go through all these industrial processes and have lots of fat and sugar and salt. So, basically it's burgers and fries, frozen pizzas, cookies, chips. I mean, everything that is good in life, they're basically saying, don't eat. But what they found, I think it was something like that the folks that had the high processed foods had about a 30% faster rate of cognitive decline than the folks who were not eating. So again, you don't have to be perfect. I mean, I had fast food today actually, but again, it's just, if you just watch what you eat, these things do add up, and can be protective. The last thing I want to mention is blood pressure, okay? So I presented this one last year, but there's an update, okay? This is the SPRINT trial. And what they did is they took, it's about almost over 9,000 people with hypertension, high blood pressure. And they either had an intensive strategy to get that, it's like 120, they want to be like 120 over 80 or whatever. So that top number is systolic, and so that's what that means. So they wanted the systolic pressure to be under 120, and then there was a standard care group where they just tried to keep it around 140 or better, okay? What they found was that there was a significant reduction in the risk of developing mild cognitive impairment or early stage dementia in the intensive strategy, that 120 group, compared to the standard care, okay? And that's the strongest evidence to date linking high blood pressure with reduced dementia risk. So again, you see the little heart and the brain holding hands. What's good for the brain is good for the heart. Put it on a billboard, done. But this is the update. So 300 of these participants actually completed a baseline and a follow-up MRI scan to look at blood flow in their brain. And what they found was that people on the intensive strategy had a significant increase in blood flow after 4 years in their brain. And the people in the standard treatment showed decreased blood flow after 4 years, okay? So, and again, those little bars, it doesn't look like, so the baseline is in the sort of the dark red, and then the 4 years later scan is in sort of that pinkish. And so it doesn't look like it's changing much, but when those little changes with respect to blood flow are important, okay? So again, blood pressure. And then building a resilient brain, and we can wrap it up. So I was trying to find a new study linking cognitive exercises with protection, and there really hasn't been much in the last year. So I'm just showing you what I showed you last year, in case we have some new folks, and just to remind those who were here last year. And so what we're talking about, we always hear about reading, writing letters, puzzles, etc. Are good for your brain and can reduce risk, and it seems to be somewhat true. This was a big study, over 2,000 participants. They were studied for 8 years, and they had an average age of 80, and they had no dementia. And those with the highest levels of mental activity, so it just in sort of a composite measure, that's the people that just did these puzzles and really exercised their brain and played cards and did things like that. They developed frank dementia, not cognitive impairment, but frank dementia, full-blown or whatever you'd say, at 94, and those with the lowest levels developed dementia at 86. So again, our lowest levels of mental activity. So again, there seems to be some protective aspect to the cognitive stuff, to being more mentally active. And what I've shown in the past, actually, and just to kind of get back to my theme, is that even people with mild cognitive impairment or early stage dementia who continue to engage in cognitive activities slow down their rate of progression. So it's protective not just for people with no dementia, but for people with early stage dementia too. Yes?
Just a quick question. Is there a difference between physically reading books, writing, playing cards, etc., versus the digital mode?
Yeah, I don't know if anyone's really analyzed that. Yeah, I mean, I know. I like to feel the book myself, but I don't know, because I think because a lot of the cognitive training is actually on they do it on an iPad, or something. So, and they have these special programs that so they're the iPad. They have these iPads that have been sort of kind of tricked out, so they're a lot easier to use. But I don't know if anyone's done a head-to-head comparison with that. Yeah. So that's all I have. That's the lab. And again, thanks as always to Miles for Memory for supporting our work. Maybe next time I'll give you an update on some things that are going on in the lab that have been supported with funding from Miles for Memory. Okay, thank you.